Hi everyone, welcome to my channel Pure Biology, and today I come with another video, that is the epitopes, and this is the topic related to the immunology. And also thank you to all of you who have already subscribed my channel, and the newcomers who please I request you to share this channel for the growth of this channel if this is helpful for you. Okay, so epitopes, what are called the epitopes? The generally the in case of immunogen, the active regions of the immunogen generally binds with the specific membrane receptor. Specific membrane receptors. Okay, for what? For continuing the antigenic reactions. Okay, or immunogenic reactions. Actually, the specific antigen generally binds with the specific antibody, not all. So, if the specific antigen binds with the specific antibody, then immunogenic reactions or activity that is continuing. So, here the two cell epitopes are, two kinds of cell epitopes are there. One is the B cell epitope, another is the T cell epitope. And here, this is the process by which the conformation of epitope that is determined. One is the crystallographic study and another is the epitope mapping. Crystallographic study and another is the epitope mapping. In the crystallographic study, there first crystals of AgAb complexes are made. First, crystals of AgAb complexes are made. Second one, beams of X-ray are passed through these crystals. Okay, beams of X-ray are passed through these crystals by which we can get three-dimensional structure. Okay, and say third one, this allowed to determine the conformation of the epitope. Okay, so first one, the crystal must be formed. AgAb complex, the crystal of AgAb complex is first formed. Then X-ray is passed through the, or X-rays are passed through the AgAb complex in the crystal form. Then that generates a three-dimensional space. Okay, and filling model or three-dimensional model, obviously. And third one is this help to determine the conformation of the epitope, by which we can easily get the actual conformation of the epitopes. Okay, or the contact residue of the antibody's antigen binding site. Okay, so here the epitope mapping, what is there? The immunogenic protein first is fragmented by any enzyme, that is proteolytic obviously, over the and that is the fragments, the immunogenic protein that is fragmented by the proteolytic enzymes obviously into overlapping peptides. Okay, and these fragments, here the fragments that are formed by the proteolytic enzymes from the immunogenic protein, these fragments, these are here the peptides are overlapping. Okay, second one, the peptides are tested. Peptides are tested and for their ability. But here the peptides are tested. How much ability are there? Okay, and here three kinds of testing are possible. One is by the native protein binding with the antibody elicited by the native protein. Second one, to induce B cell activation, and third, to induce the T cell activation. So again I repeat, what is that? This epitope mapping is what? First, immunogenic protein that is fragmented by the proteolytic enzymes into overlapping peptides. That means the fragments that containing the peptides. Now the peptides are tested. How much abilities are there in the peptides? How the testing is doing? Number one, to bind with antibody by the native protein. Second, to induce the B cell activation, and third, to induce the T cell activation. Okay. So these are the two processes. One is the crystallographic process, another is the epitope mapping process, by which we can understand the actual conformation of the epitope. Okay. So after that we will learn the features of the B cell and the T cell. So continue. So here the features of the B cell epitopes and the T cell epitopes. B cell epitopes, here the size of the B cell epitopes is determined by the size, shape and also amino acid residues of antigen. What? By which we can understand? By the size of antigen, shape of the antigen and the amino acid residues of antigen that reveal the size of the B cell epitope. Clear? Second point, the B cell epitopes in native proteins are hydrophilic amino acids. There are hydrophilic amino acid residues. Okay. Third one, these contain sequential amino acid residues. Okay, from segments to the chain. Okay, segments to the chain, it consisting of sequential amino acids. Okay, and fourth one, these tend to be located in the flexible region. And here what is the actual facility or benefit of the sequential amino acids? This brought together by folded conformation of the protein antigen. That means this actually containing the sequential amino acid. So here the fragments or the segments that form two protein folding. Okay, protein folding, because the amino acid that come close together and form the folding structure. And generally B cell epitopes, they remain in the flexible region. So these are the four features. There are many other features, so I have already only written here the four features. In case of T cells, these are the processed linear peptides. Okay, processed linear peptides, and they remain in associated with the MHC molecule. Okay, remain in associated with the MHC molecules. Second one, the size is determined by the size of same thing, antigen binding cleft of the MHC molecule. Here, by the size of antigen or amino acid residues or the shape, that reveal actually the size of the B cell. But here, the cleft of the MHC molecule, okay, the binding cleft of MHC molecule, that reveal the actually size of the cell epitope. Clear? And third one, this forms a trimolecular complex involving a MHC molecule, processed antigenic peptide and T cell receptor. What are there? Trimeric means three molecules are there. One MHC molecule, second a processed antigenic peptide, and third a T cell receptor. Three molecules combined together to form a complex. The fourth one, these consist of amphipathic peptides. That means the peptides may be hydrophilic or hydrophobic amino acid. Peptides containing obviously hydrophilic or hydrophobic amino acids. If hydrophilic amino acids are there, then these are called agritopes, or they act like agritopes. Okay, and if the amino acids are hydrophobic, then they act like the epitopes. So these are the just main few features of the B cell epitopes and the T cell epitopes. So here the actual information or the completion of the topic epitopes is clear, and I think you all understood and by this explanation. And so I will upload, or I will give another lecture on another topic in the related immunology, obviously. So continue with me and my channel, and obviously share. I again repeat, you share this channel with your friends. Okay, thank you.